For decades, Alzheimer's disease research has operated within three heavily siloed disciplines. Researchers study neuronal proteostasis, focusing on the accumulation of amyloid and tau proteins, or they study microglial neuroinflammation, mapping out the brain's immune response, or they observe the systemic metabolic decline of the aging patient. This diagram illustrates how treating these readouts as parallel, competing hypotheses has produced generations of fragmented therapeutic approaches. To understand why removing amyloid or blocking inflammation fails to rescue cognition, we have to look beneath the isolated symptoms to the shared cellular architecture supporting them. The bioenergetic collapse model proposes that protein aggregation, inflammation, and metabolic exhaustion are sequential manifestations of a single progressive collapse in the cellular quality control machinery. Tracking this failure requires a specific analytical trajectory. We have to look inside the dying neuron, examine the metabolic exhaustion of the surrounding microglia, and ultimately map these cellular events to the systemic allostatic load of the entire organism. Integrating these isolated research programs reveals four emergent convergences that unify the disease pathology. If the field is going to understand why amyloid-clearing therapies fail to preserve circuit integrity, it has to abandon the amyloid-first model. The focus must shift to identifying the exact point of bioenergetic failure that precedes the appearance of the plaque. The conventional timeline of the amyloid cascade contains a structural error. Phenomenological data originating from Gouras' laboratory at Weill Cornell demonstrates that amyloid beta 42 accumulates inside neurons long before any extracellular plaques are visible in the brain. This intraneuronal accumulation is highly specific. It is localized precisely within the multivesicular bodies and late endosomes, the organelles that make up the cell's waste disposal system. The mechanism driving this buildup was identified by Ralph Nixon. Genetic mutations associated with Alzheimer's disease directly impair the assembly of a specific enzyme complex called the VATPase. Without it, autolysosomes cannot acidify. They lose their ability to degrade and clear protein waste. This acidification failure initiates a trajectory known as PANTHOS. Uncleared autophagic cargo builds up internally, causing the organelles to massively expand, until they rupture, killing the cell and violently extruding a dense core of biological material into the surrounding tissue. This finding alters our view of the extracellular amyloid plaque. The plaque is the extruded remnant of a neuron that died from an internal autolysosomal failure, rather than an infectious seed secreted by a healthy cell. Tracing this failure back even further reveals an earlier defect. The research programs of Russell Swerdlow and Richard Ewell establish that this lysosomal acidification failure is directly downstream of an age-dependent collapse in mitochondrial quality control. Cells maintain energy through canonical mitophagy. When a mitochondrion fails, PINK1 accumulates, recruiting Parkin to tag the dead organelle for degradation. When this fails with age, damaged mitochondria accumulate. They consume resources, halt ATP production, and leak reactive oxygen species that degrade adjacent structures. This resulting drop in cellular energy output directly suppresses TFEB, the master transcription factor strictly required for the cell to build new lysosomes. The sequence is highly coupled. Mitophagy fails to clear dead mitochondria. The resulting ATP depletion and oxidative leakage destroys the lysosome's capacity to function. The lysosome fails, triggering the PANTHOS neuronal death cascade. As the PANTHOS neuron dying and extrudes its core, the investigation shifts to the microglial cells migrating to engage the newly formed plaque. Microglia arrive to execute an energy-intensive immune clearance program. This process is initiated when their TREM2 receptors bind to the lipid and amyloid cargo sitting in the extracellular space. Executing clearance requires metabolic reprogramming. The signal passes through the PI3K-AKT-mTOR pathway, attempting to ramp up oxidative phosphorylation, but as Minhee Baik demonstrates, this shift stalls. With a degraded mitochondrial population, the cell cannot meet the energy demand. Unoxidized lipid pools into gridlocked cytoplasmic droplets. This redefines the post-homeostatic microglial state. these cells move past a state of activation into one of profound bioenergetic exhaustion and failed salvage. Gridlocked, energy-depleted immune cells become engines of circuit damage through the activation of the NLRP3 inflammasome. This specific effector gate is responsible for switching microglia from failed salvage to active destruction of the surrounding brain tissue. The trigger for this switch traces straight back to failed mitophagy. As damaged mitochondria pile up inside the microglia, they lose membrane integrity. They leak mitochondrial damage-associated molecular patterns, specifically fragments of oxidized DNA, into the cytosol. These fragments cause the NLRP3 inflammasome to physically assemble. The downstream consequences of this assembly are explosive. It activates caspase-1 enzymes, forcing the mass release of highly destructive IL-1 beta cytokines and matrix metalloproteinases out into the brain tissue. Alzheimer's neuroinflammation emerges as a direct enzymatic reaction to a microglial cell spilling its own decaying mitochondrial DNA into its cytosol, rather than a primary response to amyloid alone. The Bioenergetic Collapse model snaps these 9 research programs into 4 emergent convergences. The first convergence recognizes that autophagy failure, mitochondria failure, and metabolic exhaustion operate as a single, mutually reinforcing quality control collapse spanning all cell types in the central nervous system. A failure at any node in this loop accelerates failure at every other node, driving the entire disease trajectory. The second convergence isolates the upstream transcriptional failure occurring inside the microglia. The model asserts that TGF-beta and SMAD signaling directly maintains microglial mitochondrial biogenesis capacity. It couples the cell's physical identity to its energy state. Therefore, the age-related loss of the healthy homeostatic microglial signature is simultaneously a collapse of its transcriptional identity and a raw collapse of its bioenergetic substrate. This leads to the third convergence, which resolves a long-standing debate over why failing Alzheimer's microglia randomly fracture into different diseased phenotypes. They do not fracture randomly. These trajectories are graded readouts of a single sorting variable, mitophagy competence. Cells with partial competence manage limited salvage. Cells with zero competence enter terminal dystrophy. The fourth convergence links organelle failure to the patient's cognitive decline. When the NLRP3 gate activates, it releases matrix metalloproteinases and cathepsins to break down protein lattices. These enzymes digest the perineuronal net's aggrecan mesh, stripping the protective matrix from high-firing parvalbumin-positive interneurons it ensheaths. Cognitive decline is the predictable endpoint of microbial mitochondrial failure, dissolving the brain's interneuron protection. To reverse this, we have to scale the cellular collapse up to the integrated metabolic system of the aging human body, relying on the mitochondrial allostatic load framework developed at Columbia University by Martin Picard. Targeted anti-amyloid monoclonals and localized anti-inflammatory drugs fail to yield durable cognitive rescue because they bypass the broken upstream energetic substrate. Removing a plaque does not restore a cell's ability to produce ATP or digest its own dead organelles. Rescue requires mitohormesis. This is the adaptive, system-wide upregulation of mitochondrial biogenesis and quality control, triggered through controlled metabolic stress. The interventions capable of inducing this response form a single, unified therapeutic class: NAD+ precursors to restore the biogenesis signaling, GLP-1 receptor agonists to reduce metabolic allostatic load, and targeted ketogenic therapies to force metabolic flexibility and mitochondrial turnover. Alzheimer's disease is a systemic bioenergetic collapse. Resolving it requires abandoning the amyloid periphery and directly restoring the quality control engine of the brain from the mitochondria outward.